Hello everyone, welcome back to this course in neuroscience. In the last episode, we got to know about neuroanatomy. In this episode, let's take a look at how we put together that very complex system of our nervous system. In this first part of episode 8, we are going to talk about the early embryonic stages of development, as well as the segmentation of the central nervous system. Here, we recommend a review of episode 7 before watching this episode. Alternatively, you can go back to episode 7 after watching this part 1 of episode 8. Early embryonic stages. Out of the 21,000 genes of our body, about 14,000 are involved in brain development. It is a very complex process indeed. The development of the nervous system begins in early embryonic stages. It is carried on into childhood and adolescence, and does not finish until adulthood. That's a very, very long process. Now, first, our embryo forms from one single fertilized egg. The egg then divides into 2-cell stage, 4-cell stage, 8-cell stage, etc. In the earliest stages, the cells are totipotent, meaning that they can form any type of cell in the body, and that includes other totipotent stem cells. Each stem cell is capable of forming one individual human being. A little bit later, there are more cells, and the stem cells become pluripotent. They are still very diverse and versatile. However, they cannot form all types of cells. For example, they cannot form totipotent cells. Later, cell division gives rise to the blastocyst. This is an illustration of the blastocyst. Our story of the nervous system begins with this. In the blastocyst, we have a process called the gastrulation. That means the blastocyst is divided into 3 germ layers. They are the ectoderm, the mesoderm, and the endoderm. The ectoderm eventually develops into the skin as well as the nervous system. The mesoderm develops into muscle, blood, and bone. In other words, muscle and connective tissue. And the endoderm develops into the internal organs. At this stage, the pluripotent stem cells now become multipotent. They are capable of producing many types of cells. However, they do not produce all types of cells in one germ layer. As you may have noticed, there is a label in this figure here that says neuroectoderm. That is a very small portion of the ectoderm, but that will be our protagonist in the next slide. So here, this is an illustration of an early stage embryo and its cross-section. This is the rostral end, and that is the caudal end. Here, at the ventral side, we have the endoderm. In the middle, we have the mesoderm. And on the dorsal side, we have the ectoderm. The neuroectoderm here forms the neural plate. It is a portion of the ectoderm. And below, between the mesoderm tissues, we have the notochord cord. The notochord cord is also an important structure. All chordates, including vertebrates and some other groups of organisms, have a notochord cord. For example, here, this is a lancelet. It is a kind of cephalochordata and is not a vertebrate. However, it still has a notochord cord. In vertebrates, the notochord cord is located ventral to the vertebra in the vertebrae. In vertebrates, the notochord cord is located ventral to the vertebral column. It releases chemical signals to guide nervous tissue formation. In the next stage, around week 3 or 4 of conception, the neural plate folds into the neural groove. The neural plate here forms the neural groove, which is bending downward. On the 2 sides of the neural groove, we have the neural folds. Later, the 2 neural folds on both sides fuses to form the neural tube. Here is the fusion, and we have the round neural tube. This process is called neurulation. The neural tube will later be our protagonist for forming the central nervous system. Besides, the neural crest also forms. The neural crest here will form the PNS nerves, the PNS ganglia, the melanocytes in the skin, and the bones and muscles of the face. At this stage, if neurulation fails, we have neural tube defects. These vary in severity. The most severe one would be anencephaly. Failure of closure in the rostral neural tube will cause the brain to fail to form. Then the fetus that is born will be dead because it does not have a brain. On the other hand, failure of closure of the caudal neural tube will cause spinal bifida. Anencephaly is a fatal condition, while spinal bifida is not. Spinal bifida has 3 levels of severity. For one, it usually causes paralysis of lower limbs, loss of control of bladder and bowels. The lightest type is the spinal bifida occulta. Occulta means that it is not very obvious. Here, only a vertebral bone is missing. There is no herniation of tissue. That is to say, the tissue does not bulge out of the body. In the second level, spinal bifida cystica, also called meningocele, the meninges herniate out, and that forms a bulge of meninges that is filled with cerebrospinal fluid but does not have other tissue in it. In the most severe level, spinal bifida aperta, or myelomeningocele, aperta means open, so here both meninges and the spinal cord herniate out. There is a sac of meninges filled with cerebrospinal fluid, and the spinal cord goes inside and then back again. This will cause a severe disruption of the functions of the lower part of the body. A critical factor for correct development of neural tube is the vitamin B9. If there is not enough folate or folic acid or vitamin B9 in the mother's body, then the deficiency might cause neural tube defects. That's why pregnant women are usually recommended to take extra vitamin B9 to ensure that there are no neural tube defects in the baby. In the next stage, we still have the neural tube, which is now more complete, and the neural crests, which have completely broken off from the neural tube. We also have formites. New structures that are formed are the somites. They are formed from the mesoderm. They later become the vertebral bones of the spinal column and related skeletal muscles. Here are the somites. The development of the neural tube and other neural-related structures are controlled by chemical signals. As we've mentioned before, the notal cord plays a role in this process. On the ventral side, the notal cord releases a protein called sonic hedgehog, SHH. SHH induces the nearby cells to form motor-related structures, that is, motor neurons and other related neurons. Here, this portion of the nervous system will be called the basal plate. On the dorsal side, the ectoderm releases BMP and WNT. These proteins induce the nearby cells to form sensory-related structures. This will be called the alar plate in the central nervous system. So we have on the one hand SSH, and on the other hand BMP and WNT. These have opposing influences on the cells. The cells are usually attracted by one and induced by this one, but repulsed and inhibited by the other. Alar plate and basal plate are therefore separated by the induction of these signals. They're not completely separated and mix a little bit, but there is a rather clear distinction between them, called the sulcus limitans. Later, in PNS, SHH still plays a major role. SHH concentration determines what a precursor cell will become. When there is high SHH concentration, the cell will become specialized glia for supporting the neural cells. When there is middle SHH concentration, the cell will become motor neurons. When there is low SHH, then the cell will likely become interneurons. That's how the PNS cells determine what they will become, what they will differentiate into. There are some other disorders besides the neural tube defects that we mentioned before. For example, if the mother is alcoholic, then the fetus might develop fetal alcohol syndrome, which is bad for the neural system development. Iodine deficiency is also fatal for the fetus. And infections such as rubella infection might cause inappropriate development of the nervous system. For example, rubella might cause deafness or blindness in the baby. Question time. Please try to select the correct statements. There may be multiple answers. A. The 3 germ layers are endoderm, mesoderm, and exoderm. B. Neurulation is the process of transition from neural plate to neural groove. C. Spinal bifida is caused by failure of closure of caudal neural tube and is a fatal condition. D. Alar plate is induced by BMP and WNT. The only correct statement is D. Let's take a look one by one. A. There is a misspelling in exoderm. It actually should be ectoderm, spelling E-C-T-O. In B, neurulation is the process of the closure of the neural folds to form the neural tube, which is after the formation of neural groove. C. Spinal bifida is indeed caused by the failure of closure of caudal neural tube. However, it is usually not a fatal condition. D is the correct answer. The alar plate is induced by BMP and WNT, while the basal plate is induced by sonic hedgehog, SHH. Up to the next section. Later, our neural tube will develop into the central nervous system. In early stages, there are 3 vesicles: the forebrain, the midbrain, and the hindbrain. At around 7 weeks, we enter the 5-vesicle stage. Here, we still have the hindbrain and the midbrain, but the forebrain forms 3 different vesicles. That includes 1 diencephalic vesicle and 2 telencephalic vesicles. Another structure that is formed around this time is the optic vesicle. This will later form our eyes. The developing nervous system is usually segmented into neuromeres. These were formulated by Orr in 1887 and verified by Mario Capecchi in 1989 via gene targeting in mice. He validated that there were different gene expressions in the different neuromeres, and indeed, the segmentation is justified. Let's take a look at the neuromeres in mouse brain and in human brain. In the prosencephalon, the forebrain, we have the prosomeres. There are 5 prosomeres. They are HY2, H11, P3, P2, and P1. HY is abbreviation for hypothalamus, and P stands for prosomeres. In the mesencephalon, we have the mesomeres. There are only 2 mesomeres. They are M1 and M2. And in the rhombencephalon, the hindbrain, we have the rhombomeres. There are 12 different rhombomeres. They are R0 through R11. Here is R0, then R1, and much more, and R11 at the caudal end of the brain, where the spinal cord begins. We might notice that the neuromeres are arranged in this order, or in the human brain like this. However, we may ask, what about the telencephalon? What about the parts superior to all these structures labeled here? Are the cerebral hemispheres part of a certain neuromere? Of course. They are part of the hypothalamic segment 1, HY1. So here, this part is HY1. The growth of the brain takes a turn at this place, both in humans and in mice. However, the human has a further turn to form the temporal lobe. So here, all these are also HY1 neuromere. Let's get into a one-by-one analysis of the 3 segments. First, prosomeres. HY2 is the terminal hypothalamus. HY1 is the peduncular hypothalamus. Terminal means at the end, so it is the very tip of the hypothalamus. And peduncular means related to the peduncles. The peduncles are the feet of the brain, because ped is a root for feet. The brain peduncles are where the brain connects the brain stem, and that's why peduncular hypothalamus. Next, there's a neuraxis flex. That is, the direction of our neural axis is changed. After the neuraxis flex, we have P3, which is the prethalamus, P2, which is the thalamus, and P1, the pretectal area. It was formerly considered to be part of the mesencephalon. However, we verified that it has similar gene expression to the prosomeres rather than the mesomeres, so it is actually part of the prosencephalon. The prosencephalon and the mesencephalon are marked by the expression of the gene OTX2. In the mesomeres, we have M1, which is the larger segment and includes the superior and inferior colliculi, and the smaller segment M2. As we mentioned in the slide before, the mesencephalon and prosencephalon are marked by the expression of the gene OTX2. The rhombencephalon is rather more complex. R0 is also called the isthmus. It is also formally considered to be part of the mesencephalon, but actually later verified to be the rhombencephalon. The dorsal parts of R0 and R1 help to form the cerebellum. Later, between R3 and R4, there's another neuraxis flex, though this flex is not like the previous one, which is very sharp. This flex is only a little bit milder. And finally, we get to R11. The gene expression here is like this. R0 is marked by the expression of FGF8. R0 and 1 are further marked by GBX2, while the rest, R4 to R11, are marked by the genes HOX1 to HOX4. HOX stands for homeobox. Homeobox proteins are a family of proteins discovered in the fruit fly, and they're later applied to the human brain and also found in the human brain. They are crucial genes for managing the development of the whole body. Before we get to the spinal cord, let's take a look at the cerebellum. So at the dorsal side of R0 and R1, we have a structure called the rhombic lip. During the development, the rhombic lip is there, and it provides a lot of new neurons. It is the most active area of neurogenesis in the developing of the brain. The neurons here migrate towards the backside and towards the midline, and forms the whole cerebellum. What is most amazing is that the cerebellum has about 80% of all the neurons in our brain. The rhombic lip is usually in charge of generating the granule cells, which is the type of cells that have the most number in the cerebellum. The granule cells make up about 70% of all the neurons in our brain. Getting back to the spinal cord, the spinal cord is also divided into several segments. The cervical segments, which are at the neck, the thoracic segments, which are at the chest, the lumbar segments, which are at the waist, the sacral segments, which are near the tailbone, and the coccygeal segments, which are near the tailbone as well. The cervical segments have 8 pairs of nerves. The thoracic has 12 pairs. Lumbar has 5 pairs. Sacral have 5 pairs as well, and coccygeal usually have 1 or 2 pairs of nerves. Note that spinal nerves always occur in pairs, one left and one right, and we usually count in pairs rather than nerves. Spinal cord segments are marked by the homeobox genes 5 to 13. Another type of division of the spinal cord is according to the function, or we could say the tissue organization. There are 6 major sections of the spinal cord. They are the prebrachial segments, from C1 to C4. They control the cervical muscles. The brachial segment, from C5 to T1, controls the upper limb muscles. The postbrachial segments, from T2 to L1, are preganglionic ANS neurons. They control the sympathetic division of the ANS. The crural segment, which is L2 to S1, controls the lower limb muscles. And the postcrural segment, S2 to S4, control the preganglionic ANS of the parasympathetic division. And lastly, the caudal segment, which is S5 to coccygeal 2, control the tail muscles. But obviously, we don't need to use them. This is only a rough description of the functions of different segments of the spinal cord. It is not very strict. Indeed, the function is not very clearly delimited. So we could say that the prebrachial segment is mainly in charge of cervical muscles, but we cannot say that it is only in charge of cervical muscles. That's almost all for today. So, a question time at the end. Try to state the following numbers. A. The number of brain vesicles in the earliest stages. B. A little bit later, how many brain vesicles are there? C, D, and E ask for the number of prosomeres, mesomeres, and rhombomeres. And F asks for the number of pairs of cervical, thoracic, lumbar, and sacrovertebral nerves. Please take a moment to pause and think about them. Here are the answers. There are 3 vesicles in the earliest stages of the brain. Then it becomes 5 vesicles. There are 5 prosomeres, 2 mesomeres, and 12 rhombomeres. And there are 8 pairs of cervical spinal nerves, 12 pairs of thoracic spinal nerves, 5 pairs of lumbar spinal nerves, and 5 pairs of sacral spinal nerves. That's all for today. In this part, we looked at the early embryonic stages of the development of the nervous system and how the central nervous system is segmented. In the next episode, we will take a look at the microscopic things, including neuron migration, axon guidance, synaptic formation, and common neuronal network motifs. Looking forward to seeing you again in the next episode.